Welcome to this Autism Ontario webinar, the latest genetic breakthrough in autism research. My name is Matthew Lay, and as always, I will be your host. We have a very exciting webinar today. A team of researchers at Toronto's Sick Kids Hospital, led by Dr. Ryan Yuen, who we have with us on the desk today, recently discovered wrinkles in DNA that may provide a genetic cause for autism spectrum disorder. The discovery has been hailed as a leap forward, as it could revolutionize not only the future of autism diagnosis, but other neurodevelopmental disorders as well. So we've got a panel here, and that panel is full of a number of people who we will be speaking to, including, as mentioned, Dr. Ryan Yuen from Sick Kids, Dr. Stephen Scherer, who's going to get things kicked off, Dr. Evdokia Anagnostou from Holland Bloorview Kids Rehabilitation Hospital, and Genetic Counsellor Ny Hoang. So it's a jam-packed day. Before I get started, let me review the stuff that I normally do. Submit your questions at any time. I will aspire to get you answers from our panel. We do have a formal presentation going on today, so it's a little bit different than some of the other dialogue we have, but we have lots of time for Q&A, and we look forward to getting your questions answered. If you have any issues throughout, click on the help button. Someone from the team will be able to assist you. And we do have some resources, including this PowerPoint deck, as well as a press release that helps explain some of this research and where some things stand. All right, with all that out of the way, let's get started. The first person I'd like to welcome to the desk is Dr. Stephen Scherer. Dr. Stephen Scherer, how are you today?
Very good, thank you. Nice to see everyone.
So this sounds like big news. Give us a little bit of a background on what we're going to be discussing today, the origins of this and the like.
Yeah, sure. So autism is a collection of different conditions that are grouped together under a common name. And many of you on the line know that our team for the past 2 decades have been using the most advanced genetic technologies to study the DNA from over 3,000 families from across Canada, many in Ontario, in order to try to subgroup the many different forms of autism. The idea is that if we can define specific forms of autism, having that knowledge in hand will better enable earlier identification, which is critically important for early intervention. And moreover, with these genetic targets identified, we will have entry points for the development of novel therapeutics for people who choose to use them. And the reason we use genetics as the entry point for our research studies is really since DNA factors likely account for 80 to 90% of the families that have autism. These are based on historical studies going back 30 years now. And so far, our efforts have led to the answer of the question, why autism comes about in my child or sibling, for about 10 to 20% of families in our current studies, with the number depending on the complexity of the autism in that individual and the family history involved. As you pointed out today, we'll talk about the latest findings of our genetic studies from an effort led by Dr. Ryan Yuen. He was the leader of this project from our Canadian team. And these scientific findings were published just a few months ago in Nature magazine. And for those of you who are on the line, this is really the leading scientific journal in the world. So this was a huge coup for not only the scientists involved, but really the families also. It's really an incredible success story. So just to open, Ny Hoang, who's a genetic counselor, she's going to give you an overview of kind of the introductory talk on genetics. And I think many of you know Ny because she's the genetic counselor that delivers actually the genetic findings from our research studies to the families that are involved. And then Ryan's going to talk about more specifics on the discovery itself, the wrinkles, as you pointed out. I think it's kind of a neat way to present it. And then Dr. Evdokia Anagnostou, who many of you know. She's probably seen many of the families involved who are online today. And myself will add comments along the way. And our kind of remit is to help interpret to the families why this is important to them. So with that background, I'll turn it over to Ny, who is going to give a PowerPoint presentation, which is really an introduction to the discovery and also autism genetics. So over to Ny.
Thanks, Steve. My role today as the genetic counselor on the team is to really help translate the complex genetic information that has been discovered into an easy to understand format for people who don't have a background in genetics or science. So I've put together a few slides to review what we currently know about the genetics of autism and what this new discovery is. And hopefully that will set the foundation for today's discussion with the other panelists. So just moving to my slides, hopefully you can see this. So I'll start off with what we know about autism spectrum disorder, and specifically what contributes to a diagnosis. So autism is considered a multifactorial complex condition, and I usually explain it to families using this cup model. You've probably seen it before, but here is an empty cup that is representing our chance of developing autism. And we all start off with a chance of developing autism. And it's considered multifactorial because we know there are many different risk factors that go into the background. So there are environmental factors that are at play. But more importantly, as Dr. Scherer mentioned, there are genetic factors. And we know that genetic factors plays a more important role by studying twins and how often identical twins develop autism together versus non-identical. So they're represented here, these genetic risk factors, as these blue and green balls. And you can see that overall, these genetic balls are bigger than the environmental balls. And so this is considered a complex condition because these different factors together is what fills up the risk cup. And so you can imagine that each and every one of us has some of these risk factors, but in order to have a diagnosis, you have to have enough of the risk factors to fill that cup. So this is what's considered a complex multifactorial condition. And from studying thousands of families with autism, we know that the genetic contribution is highly variable. So I'm showing you here 3 separate cups that are all filled. So they're representing the genetic profiles of individuals who have autism. And like the uniqueness in terms of presentation of every child with autism, the genetic underlying background is also very different. So in the first cup, you'll see that there is one kind of strong genetic factor, a large ball filling up the majority of the cup, where you only need a few other risk factors to reach that threshold to fill the entire cup. In the second cup, this individual's autism is due to a combination of more moderate genetic factors. So I'm showing you here 2 kind of moderate-sized genetic balls. And then in the last cup, which is actually accounts for the majority of individuals with autism, we don't find a single or a few genetic factors that are strong and moderate. So the thinking is their child's autism is due to a combination of weaker genetic factors. So genetics is still at play here, just weaker impacts. And then the environmental factors are modifying the risk. They don't in themselves fill up the entire cup. And I've listed some examples of some genetic factors, and we'll go into the different types in the next few slides. So those genetic risk factors, what are we actually talking about? So these risk factors are actually a change in our biology. It's a biological difference. So we know that the human body, males and females, we are essentially just building blocks of cells, right? Hair cells, brain cells, skin cells, heart cells, and in nearly all of the cells of our body is the same set of instructions that tell it how to grow and function properly. And so those set of instructions you can actually visualize by looking at a cell under a microscope. And they're represented here by these blue structures that we refer to as chromosomes. And so when you zoom into a chromosome and you unwind it, you can actually see the genetic instructions, the DNA sequence, a combination of genetic letters that make up our genetic code. And it's that sequence and code that is the instructions for making proteins. And you can think of proteins as workers in the body. They have very specific jobs to do, and we need them to grow and function properly. So these genetic contributions in that cup model I was referring to, they're actually representing a genetic difference. So a change in the DNA sequence that we refer to as genetic variance. And so if there's a variation in the code that makes the protein, perhaps the protein is not made the way it should be. So you can see here the protein's a bit short. And then it may not be doing the job that it's intended to do in the body. So let me give you one example. So let's take a protein that has a very important job in the brain. And I'm showing you here, perhaps the protein is responsible for a job that helps one brain cell, one neuron there, communicate with another neuron. And so let's say that the typical function of this protein is to help send a chemical signal from one brain cell to another. So it's illustrated here. Now, if that gene that makes that protein is altered, perhaps the protein is not working, and maybe that translates to the signal being sent too often, what we sometimes call overfiring, overstimulating. And the reverse can also happen, where the signal is being sent, but perhaps more slowly. So this is an example of just what could happen when you have a genetic variation and then a change to a protein. So genetic variations are those genetic contributions in the cup model, and there are different types that have been uncovered in autism. So one type you may have heard of is called deletions and duplications. And I'm showing you here an example of a deletion, where there is a segment of the genetic information that has been removed or lost, and so is shortened. Here I'm showing you a duplication, where a segment of the genetic information instructions is copied, duplicated. So you can see in comparison to the reference, it's not what it should be. You either have loss of instructions or gain of instructions. And there are a few examples that I've listed here. Sometimes deletions and duplications may involve many genes in the region. So 1q21.1 is an example of deletions and duplications that have been detected in autism, as well as 16p13.11. And then I've listed some genes where the deletions and duplications are only involving those genes. So neuroligin 3, astrotectin 2, MBD5, and PATCH-D1 are a few examples. Another type of genetic variation that we can get is called a single nucleotide variation. And this refers to a genetic change at the level of one of those letters. So you can see here, one letter of our genetic code, the G, is being altered or switched to another letter, T. And so there are different genes that have been identified to play a role in autism where single nucleotide variance is detected. So CHT8, ARID1B, and so on. So that's a different type of genetic variation as well. The last type of genetic variation I want to go through is related to our discovery. So it is called repeat expansions. And you can see here, there is a section of the genetic instructions, CGG, that's being repeated in tandem is what we call it. So it's CGG repeated 5 times here. What can sometimes happen is that CGG repeat gets expanded. So instead of having 5 repeats, I'm showing you an expansion to 9 repeats. So this type of repeat expansion has been discovered as a mechanism for many different disorders. One common example is Fragile X syndrome, which is the most common cause of intellectual disability in males, and has also been associated with autism. So the typical range of CGG repeats within this FMR1 gene is between 5 and 40 repeats. That's the normal size we would expect. When the repeat gets expanded to beyond 200, that's when we would have Fragile X syndrome. And as I mentioned, there are many different genes, between 40 and 50 different disorders actually that are linked to these repeat expansions. And I'm just showing you a few here. And there's a few important things to note about these well-known repeat expansion disorders. So one is that this disorder is linked to a well-defined repeat pattern, what we call a motif. So with Fragile X syndrome, it's the CGG repeat within this FMR1 gene that is the motif. In Friedreich's ataxia, it's a GAA repeat in a gene called FX1N, and so on with myotonic dystrophy, type 1, Huntington's disease, and spinocellular ataxia type 10. The other important thing to note is with these well-known disorders, it's typically only one pattern per disorder or one motif. And the other thing that is important is that the normal range and the expanded range for these disorders are well known. So I've listed here on the right beside each disorder what we would expect an expansion related to that order would involve. So for Fragile X, you have to have more than 200 of the CGG repeat. For Friedreich's ataxia, it's more than 100. So the number changes and the motif changes, but it's typically one motif per disorder. And we know what the typical range should be versus the expanded range. The reason I go through this is it's related to this new discovery by Dr. Yuen's lab. So this is an image from the Sick Kids press release that really illustrates what I mentioned previously about the tandem repeats. And it's showing you that in the parent who's atypical, in the parent who is typical, meaning does not have a diagnosis of autism, we see the repeat detected. And in the child, that repeat area is expanded. And so this is typically what's seen with the other disorders as well. The expansion is from one of the parents' alleles or copy. There are important differences about this new discovery, though, which I want to take the next few slides to point out. It is a lot more complex than these well-known repeat disorders. So one important difference is that for autism spectrum disorder, it's not just one gene. So I've listed here 8 genes that have been implicated in autism that have been included in the publication. So that's the first difference. The second difference is that it's not just one pattern or repeat motif. So if we take one of those genes that have been implicated in this publication, CACNB1, we can actually see that in some individuals with autism, the repeat sequence or pattern is AAG, and in others, it's AAG, AAGGAG that's entirely repeated. And the last example is a slight difference as well. So it's actually 3 different patterns that are being repeated. So this really highlights that it's definitely more complex than the well-known repeat disorders, where it's one gene, one repeat segment. And then lastly, the important kind of new thing about this discovery as well is that we don't know a lot about it yet. So the normal range of the repeated section in all of the genes has not been well characterized. We don't know, for example, what the normal size is in the general population. So we need to do more studies looking at these repeated patterns in more individuals, both with autism and also without autism, to fully understand the impact of this finding. So those are all the slides I have, just kind of set the background for the discussion with Ryan and with Evdokia.
Nice. So some absolutely incredible information. And we do have one question that I'm going to ask you, because we're going to save most of them for the end, I think, but I do want to ask you one, and that came from one of our audience members who asked, do all the cups contain at least one genetic factor? That is, you've never seen a case where it's all environmental and no genetic factors at play?
Yes, so that is correct. We expect that genetics always plays a role. Some of them are minor roles. So one of the things we're trying to look at now is what we call a polygenic risk score, where we look at more common genetic variants that have smaller impact. So you imagine them as small genetic balls in the cup, but we would never expect just a cup with environmental balls.
Okay. Well, thank you very much. We're going to be coming back to you at the end for our Q&A. A reminder for everybody, and that is submit your questions. If you're like me, this is a lot to take in, and so I'm sure that there's going to be more than a few clarification questions. We're going to get to those towards the end of today's conversation. So what I'm going to bring up now is Dr. Ryan Yuen. Ryan, this is I guess your baby to a certain extent. I know everyone was involved. Can you talk to us a little bit about the origin and comment on all that great information that Ny just provided us?
Thank you. It's my pleasure to be able to be here to share with you our latest and our breakthrough findings. Those excellent introductions already set up the background of the research, the limitation. So I just want to further explain why this is challenging. So as Ny give us a good example of a region where there's an expansion, which is the FMR1, one of the very first genes that known to be related to autism. That was actually identified 30 years ago. But then after that, the discovery, as Ny mentioned, around only 50 loci were further discovered, and not much is known afterwards whether or not those regions would be related to autism or other complex disorder. The reason is because these repetitive regions are challenging for the technology to be able to identify it. Our technology of studying DNA has been evolving and improved. One of the breakthrough would be around a decade ago of the so-called next generation sequencing. So just briefly, basically is the short sequence that trying to sequence millions of them at a time, because our genome, which is the makeup of our DNA in our body, consists of over billions of nucleotides, as Ny explained. So in order to understand or look at all these nucleotides in our genome, we need genome sequencing, and it's been recently developed or mature enough to interrogate our whole genome. Even though we can look at all this nucleotide at single nucleotide basis, the challenge is analyzing. So we have the data, but how to analyze is the problem. You can imagine it like measuring a height of a person, but you only have a ruler of this short. So how could you measure an individual's height, which in this case is more like the repeat size, with this short rulers? You can stack them and add them up to roughly calculate the size, but that's not efficient. So the way we come up is, for example, we have a room of people of different heights. A more efficient way to tell who is the tallest is to line them up based on their height. And so this is exactly what we did, what we call the outlier approach. And the tallest one, we would like to know why does it make it so high, so tall? So we asked the person, what do you do? And the person say, well, I play basketball. So you can imagine basketball would be more like autism. So that set us the hypothesis. So could playing basketball always being high in this repeat? And so we collect a large collection of individuals that play basketball versus the people that do not. And we line them up again to see if there are more people being high in the height in people playing basketball versus the ones that do not play basketball. And indeed, what we show is the case. So it's more like autism here. We line them up the repeat size, and we see that the repeat length having the long repeat length are more likely to be found in individuals with autism versus the ones that do not. So this is really the breakthrough of analyzing the whole genome of the potentially contributing factors, genetic factors in autism. And that lead us to identifying the relevant genes that associate with this repeat. And some of them, most of them, we do not know before, which again opens up a lot of new opportunities in precision diagnostics or even targeted medicine or treatments. So this is a simplified way of the breakthrough and how the method we used to discover this.
Thank you very much, Dr. Yuen. I'm going to reserve the questions, since I gave my prompt, are flying in. So I'm going to reserve that for the panel when we bring you back. But next, I think we want to take a look at the family's perspective. And for that, I am going to welcome to the desk Dr. Anagnostou and ask you to give us a few words about the family perspective as it relates to this.
So hello, everybody. I cannot give you the family perspective. I'm not a family member, but I can tell you a little bit about why families care about these types of information, or why families should care about this type of information. So when we have a new genetic discovery, there is often a question asked about how does that change what you do with families and kids. And I hear from families 3 types of benefits that they get out of knowing that their child has a particular genetic variant. And one has to do with what we call explanatory power. So lots of you tell us that it was always a mystery why their child had this particular difference and a particular set of difficulties. And there was a lot of relief from understanding and knowing what the original cause, the original etiology of the condition was. And for some of the families, it actually takes away fears about things that they may have done, they may believe they may have done that were associated with autism in their child. So to know that it lives in the blueprint, that is our genetic code, provides explanation and sometimes some relief. The second reason, and it has been already identified by the previous speakers, is early diagnosis. So we know that for kids who have autism spectrum disorders or a related condition, early intervention is very important. And we are limited in our capacity to provide early intervention by the time of diagnosis. So we need the symptoms to be obvious before we can make a diagnosis. But if we had a mechanism for identifying some of the kids who were at risk for developing autism as they grow early on, then there is the potential for providing early intervention to help with the skill building that is required to help kids and youth and adults as they grow with ASD. And the third thing that has already been mentioned by Dr. Yuen, I would say that's very important to remember, is when a family gets a genetic finding back, they typically will ask me if I have now a treatment or a therapy or an intervention, depending on their view on this topic, that is targeting, that's special to their child. So the truth is, and we have to be very honest about that although we all know that autism is a collection of different conditions, right, that does happen to share social deficits and repetitive behaviors as characteristics, when it comes to interventions, we often offer the same intervention to all kids. So most of our intervention plans do not include a consideration of the specific autism that the child has. And that's because we don't understand all the different autisms yet. But genetic information, when we get it, and a breakthrough like that, where it increases our capacity to identify the different genetic differences that are associated with autism, then starts giving us information about what specific type of autism a child or a youth or an adult has. And then it opens up the field for thinking through what is special about this special difference and how can we tailor, whether it is therapeutics in terms of medications or other types of interventions like psychosocial interventions and technology, to the type of difference that this particular child has. And this is the ultimate goal, right? The ultimate goal is to understand all the different autisms so that we can provide solutions to families and kids and youth and adults that are tailored to their own version of autism, right? So understanding all the differences in our blueprint that are actually contributing to these genetic disorders and how those affect our brains and our bodies and our ultimate development is critical to be able to think through all the different types of interventions and how they get tailored to the person's specific need. So I'm going to leave it for there, and I'm happy to take questions after.
Excellent. Thank you. Thank you so much, Doctor. And we've got a number of questions. Keep them coming. We got a lot of time, as you can see, that we shared. So we're going to bring back the whole panel now. And what we're going to do is start working through these various questions. So Dr. Scherer, maybe I could start with you on this question and tell me, everyone else can also comment on it, but how do you feel that this, I mean, it seems like a massive discovery, will influence the treatment of autism in the near future? Is this a near future impact thing?
Well, maybe I'll just begin by saying I think this is the most exciting advance in autism research in 15 years. So, and that's captured by the fact that Nature magazine published this. And it's exciting really for many reasons. And the first is that when Ny had mentioned these large circles, these high impact changes, genetic changes, we had pretty good confidence of about, I would say, 50 or so that are used in the diagnostic setting, okay? And then with this new paper that Dr. Yuen talked about, we essentially added 57 new regions in the genome. Some of them overlap with the existing genes we knew about, but when they did, we have a new class of genetic variation, as Ny pointed out, that we didn't know about before. But then the other ones are brand new. So they're new diagnostic targets and they're new therapeutic targets. This is unbelievable. It's very, very highly debated in the field how much genetics plays a role in autism. I liberally use the 80 to 90% term. Ny implied that 100% of some form. I think there's a very important question asked earlier, how much is environmental? Depends how you define environmental. I define environmental as anything that's not genetic. Okay, so it could be the food you eat, the toxins you're exposed to, how much light you have, the temperature that you're raised in. I mean, it's everything that's non-genetic. So I think the other important point of this discovery, it adds yet another level of complexity about autism. If there's one thing we know, everything about autism is complex. But at the end of the day, we add some simplicity in that we've identified some more of those large circles in these genetic model. And this is hugely important for the 3 points that Dr. Anagnostou pointed out. We will use some of these already in diagnosis, so that 10 to 20% number I talked about in my opening comments, now that's pushed up. We don't know exactly how far yet because it's very preliminary data, but I'm thinking it's going to add another 5% or so to that. So we're going to be in the 15 to 25% range. This is hugely important when you think about, one in roughly 100 individuals will get a diagnosis of autism worldwide. So it adds to the numbers. And secondly, you can use this information for early diagnosis. We have to get the process in place, and we're working on that. And third, the approach to develop new therapeutics is really based on having a target, right? If you're going to develop a medicine, you need to know what you're going to try to modulate. And that's been the challenge in autism for the better part of since 30, 40 years, is we didn't have these targets, and now we do. So things should move a lot faster. There's a long way to go, but this discovery really opens up a lot of new pathways into understanding autism and helping the families involved.
Excellent. So I think this is a question for Dr. Anagnostou, who I'm now not getting the name right after I started doing it so well. So I will continue to do my best here. But there's people that are wondering how they get involved in future studies. Is that a question that you can answer for us?
I'm very happy to answer that question. So basically, one of the take-home points of this particular discovery is that the family's investment in these research studies actually has big impact on how we understand autism and how we help these families, but also other families to come after them. So we're obviously thrilled when families want to participate in research. There are several ways people can participate depending on where you're calling from. The easiest one, if you're in the province of Ontario, which I think the majority of our audience is to get involved in the PONT network, which is the Province of Ontario Neurodevelopmental Disorders Network study. That is a study that goes pretty much across Ontario with few exceptions. And so we take, you are welcome to contact the PONT study. We can post the link with Autism Ontario with this webinar to give you access to the personnel that actually screens people for this study. But basically, we take everybody who has a diagnosis of autism. And if you're in a region where we cannot reach you, we can try to find a solution either through remote ways of collecting some data from you, or finding another study that we may be collaborating with that is in your region. So we're thrilled to hear that the families want to participate, obviously. We have created some infrastructure across Ontario to allow that, and we will put a link with this video at the Autism Ontario website. And even if you're from a place that we cannot reach you, we'll find some solution for you to contribute your genetic sample.
Excellent. So this next question, I think I'm going to direct it at Ny, but you all may want to jump in on it. What causes the genetic modifications? Is there no family history of any genetic diseases? Why do they happen during pregnancy? Ny, I'll throw it over to you first. Let us know what your thoughts are on that. Oh, I think you're muted there, Doctor. I will fix that. Okay, you're good to go.
Okay, perfect. You can hear me now?
Yes, we can.
Okay, wonderful. Yeah, I didn't touch on inherited or de novo variations, but it really depends on the variant itself. Some could be inherited, others could be new. That was kind of at the time of conception, when the egg and the sperm came together, the change happened. We often will put more significance on de novo changes, especially when the child has autism and both parents don't. And so we do see kind of a mixed bag of de novo and inherited variations. Some have more impact than others. And then in terms of the family like history or the pattern running in the family, it's not uncommon to see kind of no patterns that's suggestive of autism kind of in one generation going down to another, going down to another. But in some, we do see a sort of a condition that looks like it's running in the family.
It's a big question.
I would also like to add the relevance in
Sorry, Ryan was speaking.
Oh, I'm sorry. Let's go back to the group. I apologize.
So in our discovery, the repeat expansion are mostly inherited, but it's more complicated than just inherited from directly from one of the parents. From decades of research, the mechanism of how the repeat expanded is we know a little bit more right now, and it is in the repair mechanism where there's a glitch that making the repeat even longer from generation to generation. So we don't know what in each locus, what is the normal size, or what is the size that will lead to a change of function in a gene, as Ny pointed out in her presentation. And this is something that we need to work on. And as I said, our discovery, we skipped that part. What we did, as I said, was just looking at the tallest individuals. What we should do next would be to set up a threshold. How tall does it make it to go to the basketball team? So that's our next step.
So on that note of next steps and everything that is going on, Ny, did you have something that you wanted to add there?
Or actually, Steve has his. Do you want to go in first?
Yeah, we're having technology challenges. We can do genetics, but not technology. I just wanted to add something that I think we think about all the time, but perhaps didn't come out in the earlier comments. And it comes back to the last question. So the brain is really the most complex organ, in our body by far. And it's really amazing when you think of the genome. That's our entire complement of DNA. We have about 3.2 billion chemical letters, like Ny talked about. And there's roughly 30,000 genes kind of strung across that DNA. 75% of those genes are involved in making those proteins that Ny talked about. Their function works in the brain. Okay, so there's a lot of these, and we know that autism is a condition of the brain. So there's a lot of different genetic targets for these different variants that she talked about. And what's really amazing to me as a person, scientist who studied genetics for over 30 years, is when you compare the DNA sequence from a mom and a dad to their child, it's almost identical. There's a mixing that happens, of course, but there's only about 70.
Dr. Scherer, we seem to have had issue just then with your audio. Just give me one moment here, and I'm going to ask you to start that thought over. It just kicked in, so one moment, please. If you could just restart that thought, I think we should be good.
The last thought or the whole string?
Yeah, the last thought. Yeah, you were good. You were good. Yeah.
So if you compare the DNA from the mom and the dad to the offspring, only about 75 changes. It's really amazing. These are what Ny was referring to as de novo or spontaneous changes. And they typically occur randomly. All these tandem repeats, these wrinkles that Ryan talked about, they have a higher rate because they slip. They're kind of like Velcro. They stick together. But anyway, but the likelihood that one of these changes is going to hit a gene that's involved in brain is really high, because 75% of the genes are involved in how the brain functions, these brain cells, these neurons. So that's one of the reasons we think that there's so many different genes involved, 100 or so we know about now. So I mentioned 50, and then Ryan's work adds another 50 or so. And then also why these changes are so important, because the brain is so important. Okay, and then the last thing I wanted to say comes back also to the question about these new changes versus inherited changes. We also have a lot of clues about the biology of autism, in that more often than roughly 4 to 1, we see autism in boys versus girls. All right, and when we see inherited genetic changes, quite often they're coming from a carrier mother, because moms have 2 X chromosomes. And males, their sons, for example, would have an X and a Y. So if they're carrying a genetic change, they have a backup X chromosome. And they're kind of protected, for lack of a better term. And then if they pass the genetic variation onto their male son, then they have the Y chromosome, which doesn't give them that genetic backup. Okay, so a lot of the genes we see that are involved in autism are on the X chromosome, which is also giving us a lot of clues to how we can use that information for early identification. So we're gathering more and more clues. The last point I wanted to make, it may come to some of the other questions, because some of you know we've been working on this for a long time. I wanted to emphasize we've actually had this whole genome sequence on roughly 3,000 families for a long time. But the data is so complex. We're just teasing through it. And Ryan really is the best example, where we've been able to use research funding coming from all of you on the line and many of the agencies that support us to bring new minds and new ideas to trying to resolve the complexity of the questions that we have. So really, Ryan came to the challenge with fresh eyes and brought a new statistical, using actually artificial intelligence approaches too, to tease through this complex data and identify these 50 new balls for Ny's cup model. Is really quite amazing. We actually had that data for several years, but kind of the old guys, the old people on the line have been looking at it. We didn't come up with the right approach. So it's so important that we generate more and more data and have fresh eyes from all around the world to come and ask these questions. And what we've done is we've actually put all of this genetic data up on the internet, and for scientists who are approved to look at it, they can take their questions to the data. And I really want to comment, in particular for those of you on the line who have contributed your samples to the study. It's fueling new ideas and research around the world. We have over 200 different laboratories from, I think it's close to 20 countries now, where scientists are bringing their bright ideas, like Ryan did, to this data and asking questions of the genetics of autism. So that 10 to 20% number, he's added another 5%. It's going to keep going up and up as we get more and more bright people bringing new ideas and bringing new explanations forward. The big question in the field is, how far will it go? Will it go to 50%, 80%, 100%? We'll see.
Ny, did you have something else to add on that topic?
I was just going to go over the inherited piece specific to this repeat. And Ryan touched on it, but maybe I can take a stab at a visualization. So bear with me. So we get 2 copies of every gene, one from mom and one from dad, with the exception of the X and the Y that Steve mentioned, right? So when Ryan says that these repeats, these wrinkles, are inherited, what he's referring to is one copy comes, let's say, from dad, one copy comes from mom and makes the child. During that process, what can happen is the repeat that's, let's say, coming from dad gets expanded. So the child has the expanded repeat on one copy from dad, and then the other one intact, not expanded from mom. And we found that with the different genes that have been included in the publication, it can come from either one. And correct me if I'm wrong, Ryan, but there doesn't seem to be a preference necessarily as far as we know yet.
Yeah, it is correct. At least in the context of autism, we didn't see a difference between father or mother.
Okay, so we've got about 9 minutes left, as best I can tell, and we've got some questions about the future and what this means. And the first one, I know it's hard, to tell, but is, with what you know about this to date, are you confident more studies will provide the answers? I guess the overall answers of breaking the code. Will this be a 5-year, 10-year, 20 years? What do we think? What do you think we're looking at? Dr. Scherer, maybe I'll start with you.
Right. So that's a great question. We need more genome sequences to look at. In this study, Ryan looked at about 20,000 samples from families with autism and roughly equivalent number of other samples from the general population. We probably need to get to 100,000 or 1 million to really resolve all of the genetic factors, including those small genetic balls that he was talking about. I actually think, I forgot to say this earlier, but I really, really, really believe, and I think this will be the major impact of this study, is I think these tandem repeats, these wrinkles, and what Ryan didn't say is there's actually millions of them across our DNA. I actually think that these are going to be the major contributor to those small balls that we know nothing about right now. So to answer your question, it's going to take us about, as long as it takes us to get to 100,000 or 1 million genomes, probably 5 to 10 years to really resolve all of the genetic contributors. But the important thing is we know a lot about the big balls in the model, and they provide us good insight on the best insight on the diagnostic and the therapeutic targets. So we also have to really focus on that now.
As it relates to that, once that occurs, do we expect that we will, and this is another question from an audience member, do we expect that we'll be able to predict the severity of autism? Or is that something that is too far out for you guys to predict at this point?
Well, I think that's a very interesting and definitely worth looking at question. So far, as Dr. Scherer keeps saying that we need more samples, we need to study more genomes. The reason is because right now, each of these cause or the expansion in this loci are very rare. We're talking about around handful of individual out of 1,000. But collectively, when we looked at, for example, around 5,000 families that we study in this research, we saw collectively they involved in autism. But individually, in each locus, it is so rare that we cannot pinpoint the impact of each locus to the individual individual cases. So without a large cohort, let's say 100,000 to 1 million, without that, we cannot gather enough samples or genomes and associate with their clinical outcomes. So until then, it will be very difficult for us to say, even in diagnostics, if we can tell its correlation with the outcome. Now, but I think it's optimistic because other known disorders for repeat expansion that he also mentioned, around 50 of them, many of them have good correlation in terms of the outcomes with the size of the repeats. So it definitely has the potential, but the limitation right now is the number. And I'm really glad to hear that a lot of you in listening or watching this webinar are interested in taking part in the research, and this is really what makes it different and what important to continue making more breakthroughs in the research in autism.
Thank you. I think Dr. Evdokia has a comment that she would like to make on this topic as well.
So Ryan's answer is the correct answer. So the thing I would like to add, and I don't know why my camera keeps changing on me, but anyway, so severity is a tricky thing. So first of all, severity of what, right? So different people care about different symptoms, so different types of difficulties, and therefore when we talk about severity, it means different things to different people. But the second thing to think about is this is the place where understanding the pathway, so understanding the specific genes involved and how those interact with the environment is critical. Because the one thing that we know is that we can modify severity, right? So the whole purpose of early intervention and the whole purpose of a lot of the things that we do have to do not with obviously changing autism. Autism is a difference that's fairly hardwired early on in fetal life, and lots of people don't even want their autism changed, right? But it's about modifying severity of the interfering behaviors, of the things that are causing trouble and distress. And so we know that we have the capacity to modify severity, but the way we do it now is in a very crude way. Like we have one plan for every kid. But if we knew the biological pathways that are affected by the genetic original change and how they're affected, because remember, you go from one gene change to, let's say, 86 billion neurons that we have in our brain that talk to each other. So we have to map that out nicely. Then we can tailor the interventions that look at severity modification, which is basically what we're trying to do. We're trying to decrease the things that produce distress and dysfunction, but the trait of autism is there and it's preserved by evolution for a reason, right? So it's staying with us. But it's about the severity. The severity question is where the center of therapeutics is, where the whole purpose of therapeutics is. And we know that gene by environment is very important in that area. So understanding the gene is as critical as understanding the environment to understand that interaction to be able to modify.
Thank you very much, Doctor. And we've got time for one more question. I know there's a lot of questions to our audience that did not get answered. As we always do, Autism Ontario will aspire to get those answers for everyone. But I'm going to throw this out to everybody because it's an interesting question, and I think you all might be able to step in on. But how should an autistic person think about themselves in light of these genetic findings?
I can take a stab at it first, if you like.
Perfect.
So this is a very important question, right? So it does not, I think sometimes there is confusion between what it means to have autism or autistic identity and what the purpose of this work is, right? The purpose of this work is not to challenge identity or the trait of autism. As I said, these are traits preserved by evolution for many, many, many years, right? And they serve a purpose, like it makes good engineers and musicians and detailed people, and it has all kinds of advantages as a trait in the general population. We only worry when these traits actually start creating dysfunction. So the way we understand new genetic discoveries is, for one, to understand the blueprint that is associated with this particular difference, and then to think about, in those people who experience distress and dysfunction, does this understanding of the original biology give us insights about how to help people? So I do not think that a new genetic finding would change the way somebody thinks about themselves. There's no reason to use a genetic finding to change the way you think about yourself. But if you're interested about all the types of genetic variations that contribute to this difference, that's interesting information. And for those who experience a distress and they want tailored intervention for their distress, it gives us an insight on how to go about developing these tailored interventions. And I hope that's helpful.
It is. Dr. Scherer?
Yeah, I would just add, that was a very nice summary. I think when Ny delivers this information back to the families, and we've done so to hundreds of families that are in our study, or the genes and the copy number variants that she had mentioned in her presentation. When I use the term subgrouping in my introductory comments, when you go back and look at those people who have the same types of genetic changes, right? These deletions or duplications or these single letter changes in a very specific gene, we've now identified, in some cases, dozens of unrelated individuals who have an autism diagnosis who have those changes. And when you get those people together or their families together, they see a lot of commonalities. We use the term quite often that autism, it's like you're a snowflake. There's millions. Everyone is unique. But in fact, genetics can have a very, very important role in the type of autism. That's what we're seeing. It's in that 20% of the subgroupings we're starting to learn a lot about. We need a lot more data. But that's exactly what we're trying to do, is to work with the doctors and the families to figure out how can we can deliver that information to best answer their questions and to move forward. And everybody's going to use it in different ways. But it adds, a type of information they didn't have before, and it answers a lot of questions the families have been looking for answers for. So we're just scratching the surface. This one was a complex one. We had to present, five different viewpoints. It probably is the most complex type of genetic variation that's been studied so far. No, it's not. It is, and I'm quite sure. This is my prediction, if you will. We're going to see this type of genetic wrinkle in other genes, not in the ones that we talked about today in Ryan's paper, but in other genes in every different genetic disorder that's known. Okay? People haven't looked yet. This is the first paper, and we often make first in autism research because we have the data, because the families and the funders have been so involved. So I think this is a first. Other kind of studies are going to learn from this one. We're going to continue to learn. Really, to get back to that question that you just asked us, Matt, is how can it be used to help define the individual and help answer the questions that they have going forward?
Well, with that, we have come to the end of our time that we have allotted for today. But I feel like this is a first conversation. This is the opening salvo, and there's going to be much more, not just data, but a lot more questions from our Autism Ontario community, parents, caregivers, families, and those living on the spectrum. So hopefully we will get an opportunity to speak with you all again. I'd like to thank you so much for the hard work you've done on this research, as well as being here with us today. For all of those out there, you know the drill. This will be available on demand tomorrow for you to view. It'll be available at the Autism Ontario website. If you know a family member, caregiver, friend, uncle, whoever it is that you think might find value in this, please pass the link along. The more of you that view our webinars, the more webinars we get to put on for you. It's just the way that it works. So with that, I want to thank our panelists. I want to thank you for your participation, and we'll see you next time at an Autism Ontario webinar event.